Hi, my name is Mikhail Lebedev, and together with my colleagues from GenX and Atlas Biomed, we are glad to present the latest version of our open source pipeline for relatedness detection for genomic data called GRAPE. Classifying the degree of relatedness between pairs of individuals has both commercial and scientific applications. From the commercial perspective, DNA relatives matching service is one of the leading drivers for direct-to-consumer genetic testing market. Today, more than 50% of genetic tests are sold for ancestry prediction and DNA relatives matching products. From the scientific perspective, DNA relatives matching is an important stage during genome-wide association studies and genetic linkage analysis. Driven by the idea of open and user-led innovation, we have developed GRAPE, the first open-source end-to-end solution for relatedness detection. Let's give a brief overview of the pipeline. GRAPE is tailored to work with genomic data stored in a VCF format. It requires a single multi-sample VCF file as input. It also has a separate workflow to download required reference data and incorporates various data pre-processing and quality control options. GRAPE uses IBs as the main tool for detection of identity by descent called IBD segments that can work with unphased data. For those who already have phased data, GRAPE provides an option to run the germline tool for the same purpose. After performing the detection of IBD segments, GRAPE uses ERSA algorithm of maximum likelihood estimation of recent shared ancestry to calculate the most probable degrees of kinship. GRAPE is built using the SnakeMake workflow and is available via the dedicated Docker container. GRAPE was originally presented at the BOSC 2021 conference, and in this presentation, I would like to describe an update that has taken place over the past year. First of all, massive refactoring has been performed. Now GRAPE is a carefully polished tool that is ready for production integration. To emphasize that, we published a scientific paper about the pipeline that is now available for review in the F1000 Research Journal. GRAPE has a comprehensive README manual and a short introduction video available on YouTube that we invite you to watch and share. Also, several important features have been added. The first one is a parallel pre-processing feature that allows GRAPE to utilize multiple cores during the pre-processing stage and significantly reduce running time while working with huge amount of samples. The second feature is the so-called IBD segments weighting. GRAPE uses detected IBD segments to estimate their relatedness degree. However, the distribution of IBD segments among non-related individuals within a population may be quite heterogeneous. There may exist genome regions with extremely high rates of overall matching, which are not inherited from the recent common ancestors and reflect other demographic factors. The implication is that IBD segments detected in such region are expected to be less accurate and prone to false positive. Our new weighting approach is intended to address this problem. The key idea is to downweight the IBD segment, that is, to reduce the IBD segment length if the segment crosses chromosome regions with a high rate of matching in the background population. To estimate the IBD matching rate, GRAPE runs the IBD segments detection using the background population dataset. This dataset should not contain close relatives, or the number of relatives should be small in comparison with the overall number of individuals in the dataset. Next, GRAPE introduces a set of chromosome windows. For that, it breaks each chromosome into the regions of fixed length and then compute an overlap length between detected background IBD segments and each such region. If the total overlap length is relatively big, we assign a weight that is close to 0 to that window. On the other hand, if the overlap length is relatively small for the background population, we assign a weight that is close to 1. As a result, we get a set of windows covering the chromosomes with weights assigned to them. We call such a set a weight mask. For instance, this figure shows the weight mask computed for the individuals of East Asian ancestry taken from 1000 Genomes Project data. High matching regions, where the weight is close to 0, are marked with the blue color. Regions with a relatively small matching rate are marked with red. We compared our weight mask approach with the ERSA exclusion mask, which is built into the ERSA tool. For that, we performed a simulation of a pedigree structure using PET-SIM simulation software. The results showed that after additional parameters tuning, our approach outperforms ERSA by several percentages in high degrees of kinship. These changes have significantly improved GRAPE as a user-friendly, fast, and accurate software. It has many different parameters to control the behavior of underlying tools and algorithms to effectively control the false positive rate and to adapt GRAPE for various research and production applications. We invite you to our GitHub repository to test and make it better together. Thank you for your attention.